In the HERO team, we are interested in the mechanisms that regulate gene expression and how alteration in these processes can lead to pathological situations in humans.
We are focusing our research on the role and mode of action of a category of transcriptional regulators that are called epigenetic enzymes. This enzyme can modify components of the chromatin, such as histones or DNA, and this is one way by which they regulate genome organization or gene expression. But this is not their only mode of action. For example, they can also affect transcription by recruiting other regulators to the chromatin.
In addition, it is really important to understand how these enzymes work because they are involved in many pathologies, including blood cell cancers, neurodevelopmental disorders, as well as muscular dystrophies.
To address these questions, we use a tiny fly called Drosophila melanogaster. Of course, when you look at a fly and compare it to humans, you wouldn't bet that they have much in common with us.
Yet, besides these apparent differences, they essentially share the same body plan organization. Also, if you look more precisely, you'll find that their muscles, their blood cells, their nervous system, and so on, they share many features with our own cells and tissues.
So what is really fantastic is that you can use Drosophila to investigate a wide range of fundamental biological questions, but also to tackle biomedically relevant issues. It is also super easy to breed. In the lab, we can host the equivalent of the population of Paris just in a few trails.
And for those of you that don't really like this horrible housefly with a continuous buzzing noise, let me tell you that Drosophila are really cute and silent. On the other hand, if you're afraid of maggots, you may pass your way.
In practice, in the HERO lab, our daily routine usually starts with a good deal of fly genetics.
But soon after, you can move to functional assays and assess, for instance, the climbing performance of your flies.
Most probably, you will also need to sit at the bench for some tedious hours of dissections to prepare your samples.
However, your meticulous work will be rewarded because next, you will use these samples to identify subtle mutant phenotype using fancy imaging techniques.
Or to perform molecular biology assays and sonky genomic experiments.
Which means that you will probably end up at your computer doing quite a bit of bioinformatics. Of course, you will have time to eat some cookies or sweets to keep you motivated.
So, if you are interested in epigenetic regulation and ontogenesis, come and join us, because together
we can be heroes!